In Chapter 3, we're going to be going up the hierarchy of organization again, and we're going to start looking at organelles and then entire cells. So we need to know what a cell is and where we learned about cells. So the cell theory has a couple of tenets. The cell theory says that all organisms are made up of cells. So we know that in order to be considered being alive, you have to be made up of at least one cell. The cell is the smallest functional unit of life. What this means is that a single cell is the smallest thing that can be considered alive, right? It's the smallest thing that can do all of those properties necessary to be considered living. And all cells arise from other cells. And you might think that last one makes a lot of sense, right? If you've seen mitosis, right? Of course cells come from other cells. But this wasn't always understood. So it was once thought that living things could come from non-living things, right? That's called spontaneous generation. Or that living things could come from different things, right? So it used to be clear that if you left meat outside, which meat is dead, right? It's not living anymore. It would spontaneously generate flies, right? And maggots. And you and I know that that's silly. So Francesco Redi in France did a experiment to see how flies spontaneously generate from meat. And he did 3 experiments, one where he set meat out in an open container, and as he expected, he got maggots on the meat. And then he put another one out in a closed container, and if the maggots were coming from the meat, then they should have appeared inside of the container. However, there were no maggots. And then he did a third one where he put meat out, but he covered it with a gauze or something that would allow the smell to come out, but no flies to get in. And what happened is the maggots appeared on the gauze. And so that was when they kind of disproved spontaneous generation. It wasn't that maggots were coming from meat. It was that the flies smelled the meat and they came to lay their larvae on a good food source. Cells come in all shapes and sizes. So there are tons of different cells in the human body. Cells are typically small. What this means is that the outside of the cell is big relative to the inside of the cell. If a cell gets too large, then the surface area to volume ratio gets too small, and the cell is not able to function. We're going to talk about some of these types of cells as we move throughout the course, but I really just want you to be able to see here is the variety, right? Not all of your cells look like the circles that we're going to see throughout this chapter. And our beginning of understanding of cells started with the invention of the microscope, right? So the first type of microscope was a light microscope. And a light microscope works by sending light through some lenses that help magnify an image, and then that image and that light is sent to your eye. So what I have here on the bottom is 3 pictures of red blood cells, so you can kind of see how they look differently in the different microscopes. So the center image here is a light microscope image. The green cells are red blood cells, and then these kind of large purple spots, those are white blood cells. And it's not a very close-up image. A light microscope only magnifies down to about 1 micron, much smaller than that, and you can't see things with good enough resolution. There's another type of microscope called an electron microscope. And the electron microscope works similarly in that instead of bouncing light off of an object, it bounces electrons. A scanning electron microscope bounces them off of the surface. So what you do to get this is you would take a sample, it actually gets painted in a metal alloy, and then you send a beam of electrons at it, which then bounce off and make an image. And you end up with something like this image on the left. So these are red blood cells right here. And you get to see kind of the surface topography of each cell. A transmission electron microscope is going to take teeny thin slices of a sample and then send electrons through the sample, right? Transmission means through. Trans means across. So a transmission electron microscope is going to look like this image over here on the right. So this is one single red blood cell right here. It's kind of bent. It's in a teeny capillary. And so this one red blood cell right here, and you can see the inside of that red blood cell. So scanning and transmission electron microscopes are really great for seeing really tiny stuff. Unfortunately, the process of preparing those samples does kill it. So the only way we're able to view something living is by looking at a light microscope. So take a moment and reflect on how the invention of the microscope could have led to the development of the cell theory.